Hello and welcome back to Paleocast. I'm Dave Marshall and you're listening to episode 95 on plants and atmosphere with Professor Jennifer McElwain of University College Dublin. Liz and I were both very fortunate to have the opportunity to grab an hour of Jenny's time while she was visiting the Life Sciences building at the University of Bristol. We never got the opportunity to present Jenny with a list of questions beforehand, like we do with most interviews, so this is just her talking from the top of her head, which I'm sure you'll agree is incredibly impressive. But what I'm hoping you'll find equally as impressive is the next episode of Paleocast, in which I'll be presenting my report from my fossil hunting expedition to the Canadian Arctic. That's more of a blog post though, and it will rely heavily on video and photos, and so therefore we won't have our usual audio episode. Around this time we should also be releasing our recordings of SVPCA, so keep an eye out for both of these on our Facebook, Twitter and YouTube accounts. All of our content is also posted to our website, where you'll additionally find extra content, such as the pictures to accompany each episode. And whilst you're there, you can also tell us what you think of the show in the comments section at the bottom of the page. And as usual, please like and share episodes on social media, subscribe and donate to the show, and send in your requests for future subjects or potential interviewees you'd like to hear from. We hope you enjoy this episode. Hi Jenny, thank you very much for joining us today.
Hi, good to be here.
Can you tell us about your route into paleobotany?
So my route was a degree in botany in Trinity College Dublin in Ireland, and in my fourth year dissertation project I was working on stomata and carbon dioxide already as an undergrad, and I read a paper of Bill Challiners, who is a paleobotanist at the University of London, and I wrote to him and to see could I do a PhD with him, and that led me to do a PhD at the University of London and focus on to bring in the geology into my training.
And what would you be doing if you weren't a paleobotanist?
Oh God, I don't know.
Out of anything in the whole world.
All the options.
Okay, of all the options, can I say botanist? No.
No.
Okay, I think I might be a mad potter.
A mad
making ceramics. I think.
Okay.
That's what I think I would be doing.
So I guess most of your work is kind of paleobotanical proxies and looking at Earth's past climate. Can you explain what this means and why these proxies are necessary?
Yeah, okay. So a proxy is an indicator.
Mm-hmm.
So it's a method which indicates something of use that we want to measure. So the proxies I'm interested in are any aspect of plant biology that gives us an indication or a measure of the atmosphere or climate in the past. So I particularly focus on morphology, so shape and structure, anatomy, and chemistry. And I try and develop proxies or indicator methods of atmospheric composition, because I'm really interested in the evolution of our atmosphere. How has it changed through Earth history?
So if I can ask you a few questions about that. If I think back to school and think about the biology lessons, and when we look at plants and what kinds of adaptations they have to their environments, so things like the number of stomata and the shape and size of the leaves and how waxy the leaves are and stuff, are those the kinds of proxies that you're using?
Yeah, absolutely. With a proxy, the more simple, the better, in a way. And if you can find a direct relationship between some aspect of the plant and some aspect of the environment, the simpler the better, because it means there's less opportunity for errors or other factors. So I focus on stomatal density, so the number of stomata on the leaf surface, and stomatal size, because both of those parameters together, they set the capacity of a plant to diffuse gas into the plant body and to lose water out of the plant body. So they're very simple. We can observe them using microscopy on fossils, but I suppose from an engineering perspective, they're absolutely crucial because they define the maximum limits at which a plant can get carbon, which is essentially food, to go through the cycle of photosynthesis.
Can I just ask a quick question? This is something I was thinking about earlier, was when I think about proxies and climate kind of proxies, I had no idea that there were plant proxies. Like most of the stuff I've thought about have been looking at, ocean chemistry and all of these kind of things. So how long have we been able to use plants as a proxy for kind of climate change?
So I would say there have, since the science began, there's always been plant proxies, and it really comes down to the fact that plants are immobile, so they can move at a generational level. So new seeds produced and can migrate a whole population, but an individual, the plant is immobile, and it's immobile in a changing environment. It changes morning to night, night to day, and it changes seasonally. So plants have to be very plastic, and they have to be able to literally change their behaviour, their physiology, within minutes, hours, and then seasonally. And the fact that they're immobile means that they're wonderful proxies. So if you can understand how they're adapted to the prevailing light in the day, or the prevailing season of a year, or the mean annual temperature, then you can use that information as a proxy. So for example, going back, something really simple like the presence of rings in wood is an indicator for seasonality. So right back to the origin of the earliest paleobotanists.
I never really thought of that as a proxy. It's just like not something I ever thought about. Yeah, it makes sense.
So those are some of the obvious physical characteristics, but you've also mentioned chemical proxies. How does the chemistry of a plant change according to the atmosphere that it's in?
So what we particularly focus on are isotopes, and we look at the two stable isotopes of carbon. So there's a carbon-12 and carbon-13, and plants actively take up the light isotope of carbon and discriminate against the heavier isotope of carbon. So you can use a measurement of the ratio of these two isotopes in a fossil. It will tell you something about every aspect of the plant biology when it was living before it became fossilized. So it'll tell you whether stomata open, whether stomata closed, did it have a fast enzymatic rate, was it in tune with the climate, and so we use carbon. It's called carbon isotopic composition, and we use this as a proxy for carbon dioxide. And this was a method developed by another paleobotanist, Hope Yaron, and Brian Schubert. Carbon isotopes have also been used to look at seasonality. And then you can look at more simple chemistry. So you can look at things like carbon to nitrogen ratio, and that will tell you something about the leaf economics. And you can even resolve that or measure that in the fossil record, and it's relatively unaltered. So if you understand what the carbon-nitrogen ratio was of the fossil, you can tell how tasty it was. Did an insect have to eat huge quantities of it to get nitrogen, or if it was really rich in nitrogen, perhaps not so much. It'd also tell you about degradation processes. So when the leaf falls, could the nitrogen be recycled quickly through the system, or more slowly, like a conifer forest today? It could take 2 years to break down that leaf, whereas a tropical deciduous leaf would go in 2 weeks. So we can use the chemistry to tell us something important about the biology, physiology of the plant in the past.
Well, it sounds like it can tell you a lot of things. Do you find that the chemical proxies are overtaking the physical proxies in terms of what they can tell you? Are they more useful?
Yeah, that's a good question. No, I wouldn't say. I think the best approach is to use both, because often if you don't, in order to study the physical proxies, like the number of stomata on the leaf surface, you need well-preserved cuticle. And in some fossils, even though there's a black carbon film, which is the leaf, it's colified, the cuticle isn't preserved, and if there's no cuticle, then you can't use the physical proxy, but you can scrape a little bit of that carbon off and look at the chemical proxy. But all methods have their weaknesses, so we think probably the best approach, like most scientists, is you approach the question with two different methods, which are as independent as possible, preferably properly independent, and they're, let's say you're trying to reconstruct CO2 using two different methods. We look at where those two methods overlap, and we report the consensus CO2, as we see that's more robust than going with one proxy or the other.
And I guess a lot of these kind of characteristics you've been talking about are all sort of on a very small scale. But is there anything that you can look at for responses to climate on larger scales, like in an entire sort of forest or entire biome, or is it all sort of small scale?
Yeah, so we I suppose a biome scale, that's really interesting. We're interested in when did the biomes we're familiar with today, when did they originate? Mm-hmm. So has there always been a tropical biome? You know, has there been a tundra biome all the way through Earth history? So if you're looking at the origin of a biome, you're looking at productivity. You might use stands of fossil forests and reconstruct their biomass. Mm-hmm. The presence of a forest in the high Arctic in the past would mean there was no tundra biome. The whole global climate was too warm, so you don't have this low diversity, non-woody vegetation at the poles. So yeah, you can get a lot of information from the individual, but then you can use different types of information to try and look at the biomes as a whole and how they've waxed and waned through geological time.
Yes, that's interesting thinking, like the difference, obviously if you have something in the high Arctic, then that means tundra's not there, and looking at the difference, kind of the gradual change from one to the other. Yeah,
and actually if you look through Earth history, the tundra biome is probably one that is least represented throughout Earth history. So between the Carboniferous Great Glaciation and the Quaternary Glaciation, that whole record, you have wooded forests going right up to the poles, North Pole, South Pole. So we have actually a cool temperate or even warm temperate vegetation at those high latitudes, and there's no tundra. So tundra is very much it's an ice house world phenomenon. So that's one of the most threatened biomes, in the future, leading into human climate change.
Well, make the most of it while you can.
Yeah.
Go see some polygonal tundra.
Go to Canada.
We can see that the physical changes in plants as a response to changing climates. But do we ever see it from the opposite way round, that the climate changes in response to what the vegetation is doing?
Yes, absolutely. So that's why plants are so wonderful, because, for an example, plants influence the hydrological cycle. And the statistic is phenomenal. Something like, I can't think exactly what it is, but a half of the whole world's water budget travels through the stomata of tropical plants every year. So plants respond to changing atmosphere and changing climate, and their physiological response can then feed back on the climate system and alter the hydrological cycle and fire, for example. So our studies at the Triassic-Jurassic boundary, we show that the number of stomata on leaf surfaces is halved. You can then model, how much water is being lost through transpiration from that vegetation, and you see that it's also halved. And if you halve the amount of water that's travelling through or moving through vegetation, but it's raining the same amount, the equation has to balance some way, and the way it balances is you get more runoff. So plant responses to climate on land influence how much water is being lost from land into the oceans. So you can actually link in the geological record, things like oceanic anoxia, eutrophication in the oceans, back to plants forcing of that climate. So plants are kind of the conduit between the atmosphere, the geosphere and the hydrosphere. So they really have this incredible capacity to alter climate. And for me, that's not so surprising if you think of the biosphere. 99% of the terrestrial biosphere is plant biomass. So it's not surprising at all that plants have a capacity to actually change climate locally or even at a global scale. Plants are cool.
Yeah. Going back to the proxies a little bit, is there any way of testing whether or not these proxies are actually valid or accurate?
Yes, and that's a really important question, and it's something that we have to do. So there's a number of ways of testing them. So you can test them using historical data sets. So we can look at the plants collected by the Victorians when carbon dioxide levels were 100 parts per million lower than today, and compare them over time as carbon dioxide has increased. And there are wonderful historical archives all over the world in museums and herbaria. So that's one way. And another way is doing it experimentally. So growing plants in a modern atmosphere and then simulating an atmosphere of the age of dinosaurs, the Mesozoic, or the year 2050, or the end of the century. And you can, even though you're only observing within the lifespan of the plant, you can't really look at genotypic change unless you look at the seeds of those plants. You can observe how the plants respond within weeks or months under these controlled environment conditions.
In these growth chambers then, what variables are you able to account for?
So when you're doing an experiment, it's really important that you only that you control everything and you only let the factor that you're testing vary. So if we're doing a CO2 experiment and we want a modern atmospheric CO2 in one growth chamber, and we want a Mesozoic atmosphere in another chamber, we have to control everything else that plants will respond to. So we control the light, the quantity, the quality, you can control the spectra, you can control the day-night cycle, even dawn and dusk and the changing light. Then you want to control temperature, humidity, watering, so precipitation, and obviously the soil medium that the plants are growing in. So you have to rigorously control everything else. And to do that properly, you need very good plant growth chambers, and you need good sensors, and you need good control between how what the sensor detects and how the controller then actually modulates or controls that environmental variable.
How long do you run these experiments for?
So it depends on the plant. So if you're working with herbaceous plants, you could run them as short as 8 weeks, but typically we work with these really ancient woody plants which are very slow, so we would run them for a minimum of 6 months and ideally a year. But with a stomatal density, we know that if you expose a plant to high carbon dioxide, the new leaves that develop already have a new stomatal density which reflects the high carbon dioxide level. Not in all plants, but in many plants. So we know that plants are responsive within the lifetime of the next leaf that is developed.
So you mentioned carbon dioxide, but what about oxygen?
So we're also really interested in oxygen. It's such an important, obviously, constituent of the atmosphere. We don't have a really good proxy for oxygen. We have great proxies for carbon dioxide. But one of the proxies that is available is the presence of charcoal in the fossil record, so it's called fusane. And if charcoal is present, that means you have wildfire, and an essential component of wildfire is oxygen. So we did a lot of experiments, probably about 5 or 6 years ago now, with a postdoc at the time, Claire Belcher, who's now a professor in Exeter. And we asked the question, what's the lowest oxygen concentration which would enable you to start a fire? So we had great fun trying to burn material, the most flammable wood known in the modern record, candles, moss on hot plates. And what you find is that 15% oxygen is the minimum concentration at which you can start a fire, and 17% is the minimum at which you can propagate and maintain a fire. But when we were doing these experiments, the plant growth chambers were very new, and I was very worried about them, and I was worried about the smoke and that it would influence the instruments. So Claire, she engineered a hoover that was in the lab, and it was the most low-tech piece of equipment, but she basically hoovered up the smoke so that none of the smoke went into the chambers. And she was working with €1 million chambers and tens of thousand euro sensors, but the hoover was the essential piece of equipment.
Was the most important.
So how far back can you use these proxies? So surely not all the plants that have ever existed have the same kind of anatomy as they do today.
Yeah, so we can use the proxies all the way back as far as we have a good terrestrial fossil record. So we can go right back to the early Devonian. But what we have to do is we have to make the assumption that plants today which are related to those fossils present all the way back in the early Devonian have a similar function or have retained some functionality of their ancestors. So, and it's very important as well, we're discovering that if you develop a calibration data set for a proxy, if you base it on an angiosperm, you can't use it universally for all extinct and or all fossil plants, even if they have a living representative, because different evolutionary groups we've discovered behave in different ways.
Is there anything that can be done to correct these problems between angiosperms and non-angiosperms?
Yeah, so you can do that, and the way we've done it is to grow lots of evolutionary different groups in plant growth chambers, subject them to simulated paleoatmospheres, and we then test how responsive they are in terms of their chemistry, particularly their chemistry or anatomy, to CO2. And what we find is that different evolutionary groups respond by different magnitudes and different rates. So what you can do is we could develop calibration data sets for every single plant, but that's not reasonable. So what we do is we can impose what we call a phylogenetic correction factor. So it's a simple factor that you can correct one evolutionary group's calibration curve, and then it can be applied to another evolutionary group for which you have a fossil.
So can we see any correlation between a different atmospheric composition in the past and the relative success of each different group at that time?
Yeah, that's such an interesting question, and we've been working on that for the past 5 years. So, and what a lot of the work is unpublished, but what we found is that different evolutionary groups have inherently different biology, and under some atmospheric compositions they outperform the other evolutionary groups in terms of their photosynthesis or in terms of how they conduct gases for photosynthesis, and there's some really surprising finds as well. So I suppose I would say, keep looking at what we're publishing, but without revealing too much. The vegetation of the Earth today is supremely adapted to our atmospheric composition, but this vegetation today would not be represented by the same numbers, the same proportionality of different evolutionary groups if we change the atmospheric composition. So I'd say in the future we are going to see big evolutionary, potentially, and ecological shifts similar to what we've seen in the Mesozoic.
Just to summarise, if you were to speak to anybody interested in studying kind of paleobotany, what would you say is kind of the significance, and how would you get somebody into paleobotany and why you should study it today?
Well, I think there are so many reasons, but the two key reasons would be, number one, it's the sheer excitement of discovery. How has this planet evolved, or how has vegetation changed through Earth history to give rise to the vegetation that we're so familiar with today, that we rely on? And that sheer discovery of understanding where the vegetation has come from, what was present, what species were present, what species went extinct, what new species originated. On the other side, I think, on a more applied side, we have this archive in the fossil record of plants responding at species level, individual level, biome level, to natural global warming events in the geological past. And I think there's so much to be learned from that record of how plants have responded to climate change and atmospheric change in the past, which it has definite relevance for helping predict what's going to happen in the future. Even though our current climate change is happening at a much more rapid pace, we can still test a lot of our ecological models or our population models today by looking at the geological past. And so I think it's those two reasons that I would encourage people to pick up a fossil plant and try and study it.
Well, Jenny, thank you very much for coming on the show today.
Thank you.
Paleocast was brought to you by Dave Marshall, with Joe Keating, Laura Soul, Liz Martin-Silverstone and Caitlin Colery. It was made possible by funding from the Paleontological Society and the Paleontological Association, but the show now relies on contributions from you, the listeners. If you've liked this episode, please consider donating, and thanks to everyone that's helped out so far. We'd also like to thank the Ocean Collective for use of their music. Please visit paleocast.com for the supplementary material to this episode and for our archive of past programmes. Subscribe to our Twitter feed @Paleocast and like us at facebook.com/Paleocast to get all the latest news.